In this video, we're going to go over oogenesis, and then we're going to discuss the differences between spermatogenesis and oogenesis. Oogenesis is the production of egg cells, or ovum. It begins during gestation, before the female has been born from the mother. You start with the stem cells for the oogonia. So this is one stem cell, so this is an oogonium. This oogonium cell is going to undergo mitosis. When it undergoes mitosis, it's going to form two identical daughter cells. One of them is an oogonium that, as a stem cell, is going to go ahead and replenish the population and essentially be able to repeat this process over and over again. The other cell is the primary oocyte. The primary oocyte is going to undergo meiosis to eventually form the ovum. Now, the interesting thing about females is that the process of forming the ovum is disjoint. It does not all happen at once. So here, the primary oocyte is actually going to start meiosis 1. And when it starts meiosis 1, it's not going to be able to complete the process. It's actually going to be halted in the middle of meiosis 1. And specifically, it's halted during prophase 1. So once the female is born, the female has a bunch of primary oocytes that are all halted at prophase 1. Another important thing about females is that this process of forming primary oocytes and the oogonia renewing itself only happens during gestation, which means once the female is born, whatever number of primary oocytes it has, that number cannot increase. In fact, that number actually decreases as the female grows just from the natural death of some of these cells. So these primary oocytes are going to be halted in prophase 1 until the female enters puberty. So starting at menarche, the first menstrual cycle, every month or about 28 days, several of these primary oocytes is going to complete meiosis 1. So this is a primary oocyte. And this process here is meiosis 1. Now, the interesting thing about meiosis with primary oocytes is that they don't produce two identical daughter cells. So one of the important things here is that when oocytes divide, there is unequal division of the cytoplasm. On the top here, you can see that we have a very small cell. This very small cell is called the polar body. The polar body is essentially just a packet of DNA. It's just going to be discarded. The other cell that's also haploid is the secondary oocyte. The secondary oocyte is the one that is capable of continuing meiosis 2 to form the ovum. Alright, so again, this is the primary oocyte. It is diploid. Through meiosis 1, the homologous chromosomes are separated, so we end up with two haploid cells. Through unequal cytoplasmic division, the polar body ends up with very little cytoplasm and is essentially just a nucleus with DNA. The secondary oocyte retains the majority of the cytoplasm, so the secondary oocyte will then proceed to meiosis 2. But here is where the process again is halted. So the secondary oocyte is going to start meiosis 2 and here this process is going to be halted during meiosis 2. Or, sorry, meiosis 2. So every menstrual cycle, a number of the primary oocytes will complete meiosis 1 to form secondary oocytes, and these secondary oocytes will begin meiosis 2 and be halted during metaphase 2. Now, if fertilization doesn't occur, then these secondary oocytes will just be shedded and they'll be gone. Fertilization is required for the secondary oocyte to complete meiosis 2. So this is the secondary oocyte that is trapped in metaphase 2 of meiosis 2. And if a sperm comes along and fertilizes the secondary oocyte, then the secondary oocyte will complete meiosis 2. And again, we have a similar situation. Meiosis 2 involves unequal division of cytoplasm. So again, we're going to form a polar body. So this polar body, again, it's essentially very little cytoplasm, really just a nucleus with DNA that's going to be discarded. You also have the other cell, the other daughter cell, where the 6 chromatids have been separated. This daughter cell is the ovum. And this ovum is then going to be able to fuse with the sperm to form the zygote. Okay, so this is the process of oogenesis in females. Again, the process is disjoint and happens at different phases during the life of the female. Gestation, puberty to menopause on a monthly basis, and then fertilization if that process occurs. So last thing I want to discuss then is some of the differences between spermatogenesis and oogenesis. So first of all, one thing we should know is that the ovum is the largest diameter cell in the human body. All right. Now, part of it helps from the fact that the unequal cytoplasmic division means the ovum ultimately gets most of the cytoplasm, but overall, the ovum really is a very large cell. It is significantly larger than the sperm cell. The sperm cell has a particular structure, whereas a cell body, which is considered the head, and then it has a flagella. And the flagella is important for motility, so the sperm is capable of moving through the female reproductive tract. In addition, on the head of the sperm, there is a special organelle called the acrosome. The acrosome contains special enzymes for breaking down the outer membrane of the ovum. And that's important because the ovum has this glycoprotein membrane shell called the zona pellucida, and it's not easy to break through. So the sperm, with their acrosomes, they're able to initiate the acrosomal reaction where they release these enzymes to break down the zona pellucida in order to get access to the inside of the ovum. And finally, another difference that's important is the production of sperm versus eggs. So spermatogenesis, as we've discussed, it starts at puberty and it continues until the male dies. So just constant production of sperm. For females, that's not the case. The stem cells, the oogonia, they stop renewing before birth. So essentially, whatever oogonia and primary oocytes there are, that number is limited once the female is born, and no additional oogonia or primary oocytes can be generated.